Hello, good afternoon. Happy Wednesday, everyone who's joined the call. I'm Pat Furlong. I'm from PPMD. I'm part of the PPMD team. And we're here today with Gerard Merrick. Dr. Merrick is with Astellas, and he's going to talk to us about the Astellas clinical trial. You perhaps already know that this is a trial really that works on the energetics of the cell. And with that, we're going to take, Dr. Merrick is going to take us through the slides, and then we'll come back and have a discussion and clarify some different things as we go along. So with that, I'm going to turn it over to Dr. Merrick.
Thank you very much for this opportunity to present our update on our program in Duchenne muscular dystrophy. Next slide, please. And just in terms of disclosures, the investigational medication I'll be talking about, ASP0367, was previously known as MA0211 and was developed by MITOCHONDRIA, which was acquired by Astellas back in 2017. And the ASP0367, as I said, is an investigational drug, which means it's not available for use outside of current ongoing investigations. The safety and efficacy of ASP0367 has not been established, and there's no guarantee that ASP0367 will get regulatory approval or become commercially available for the uses that we're investigating. And ASP0367 is not authorized for sale in any jurisdiction at this time. Next slide, please. So mitochondria, for the overview, we're going to be talking a little bit about mitochondria, which are important parts of muscle cells, important parts of every cell. Mitochondria, what they do is they make the energy that your cells need to survive. And in Duchenne muscular dystrophy, there's downstream, there's problems with mitochondria that are downstream due to the loss of the dystrophin. And as a result of that, the mitochondrial function is impaired. ASP0367, our investigational drug, is being tested to find out if it relieves some of the key symptoms of Duchenne muscular dystrophy. It's given by mouth and may be given in combination with approved therapies such as exon skipping treatments and corticosteroids. ASP0367 may act inside muscle cells to increase the mitochondria's ability to make energy. And we are testing it whether that will translate into improving your muscle health and function. And a phase 1B study is open now in the United States. There are plans to open several other sites soon. The sites that have opened include Sacramento, California, Kansas City, Kansas, Worcester, Massachusetts, Norfolk, Virginia, Richmond, Virginia, and not listed on this slide deck is also included in Minneapolis, Minnesota. Next slide, please. Next slide, please.
It's up.
Oh, I'm sorry. My slide seems to be hung up here. I can, I'll just bring up my own slide deck and then.
Yeah, that's fine. So your
okay.
The slide that's up is slide 4?
Yes, yeah. So I just want to tell you a little bit about Astellas. Shown up here is a picture of a corporate office, development office here in Northbrook, Illinois. The corporate vision of Astellas, however, is to be on the forefront of healthcare change to turn innovative science into value for patients. So here's some quick facts. Astellas was formed back in 2005 from the merger of two Japanese companies, Yamanouchi and Fujisawa, and it's headquartered in Tokyo with locations worldwide. There's a growing expertise in areas of discovery research, including regenerative medicines, muscle disease, and neuroscience, and a conscious company a shift to a focus area approach. We try to take a science-first approach, empowering the best science, best talent, and developing these projects in the best locations. We have a broad pipeline, primarily focused in areas like oncology, urology, nephrology, and immunology, where there is high unmet needs, such as products that in the past, if you have someone that has had organ transplant, you've probably been on Astellas drug there as well. In terms of the numbers, Astellas is a top 25 global pharmaceutical company, employing more than 6,000 employees worldwide, with over 2,000 working in M&D. The revenue is around $11.8 billion annually, and we convert about 16 to 20% of our sales year to year is reinvested into R&D. Next slide, please. I want to say a little bit about mitochondria, which I talked on initially, because this is actually what target of what our drug acts on. And shown on the top left is a picture of muscle cells, and then mitochondria are actually present within the muscle cells. And the insert on the right is sort of a very high-powered view where you use microscopes like electron microscopes to go down basically to a cellular level or subcellular level in the case of mitochondria. So mitochondria are very small parts of your cells that have a very specific job. They turn the food that you eat into energy that your cells need to survive and do work. And there are special pathways that regulate how active the mitochondria are. How the mitochondria allows the cell's energetics to act, how they use sugars and fatty acids that are the smaller sources of energy from the food that you eat, and how many of the mitochondria that there are. Now, you all know that dystrophin is a protein inside your muscles that works with other proteins to keep your muscles strong and protect them from injury. But when your muscle cells don't have dystrophin, they get damaged over time by calcium leaking inside them. This also weakens the mitochondria. And damaged mitochondria can't use fatty acids properly. And when this happens, your muscles get tired easily. They can't repair injuries from being used, and they get sore. Next slide, please. So in terms of ASP0367, I just want to describe on a basic level what it's doing. It's being investigated to determine if it can turn on the PPAR delta pathway. So PPAR drugs have been known for a while and are used in other areas such as diabetes. We have a somewhat different PPAR drug affecting the delta pathway. And this PPAR delta pathway regulates mitochondria by turning on different genes in the cell. When the pathway is on, the mitochondria use fatty acids more, and more mitochondria are made. And using fatty acids for energy results in increased energy production and improves your muscle health and function. So these mechanisms may allow testing the combination of the use of ASP0367 with exon-skipping therapies and or corticosteroids. Next slide, please. So for ASP0367, we're going to be testing possible combination therapy actually from the beginning. ASP0367 is being investigated whether it can turn on the PPAR delta pathway and then improve your muscle health and endurance. And some drugs, some approved DMD drugs act upstream of ASP0367 at the DNA or RNA level. And the PPAR delta pathway occurs downstream of mutations in DNA and RNA. The concept for ASP0367 is to use it as a treatment to relieve the symptoms that would be used on top of other therapies such as exon-skipping treatments and corticosteroids. Astellas is interested in testing whether ASP0367 might not only be safe to use, but also be increased muscular endurance. So in this trial, subjects can enroll if they are on a stable dose of an approved exon skipping drug and or corticosteroids. Next slide, please. So this is the title slide for our study that has started, a study to assess the safety, tolerability, and preliminary efficacy of ASP0367 in pediatric male participants with Duchenne muscular dystrophy. And the clinicaltrials.gov number associated with this is NCT 04184882. And so as you know, you can look it on clinicaltrials.gov using this number, or you can actually even punch in the drug number, ASP0367, and you can find this currently ongoing clinical trial that we have. I should mention we've already tested the safety tolerability of this drug in healthy volunteers, allowing us to move on to this study. Next slide, please. Slide 9. So just describe, this is the phase 1B clinical trial that we have ongoing now. It's randomized. It's double-blind, meaning neither the investigator at your site, the PI, the principal investigator, or you will know whether or not you're on active drug or placebo. And placebo, as you know, is like the active medication, but it doesn't have the active ingredients in. So it's sort of like a sugar pill. So this is a controlled trial, though. And the primary purpose is to evaluate the safety and tolerability of ASP0367. The study will also evaluate how the body processes the drug, namely drug levels in the blood, and also how the drug affects function with an arm cycling test that I'll describe in a little bit. And as I said, trial sites are opening now in the United States. And to find a participating site near you, you can visit clinicaltrials.gov and search NCT 04184882. And as there'll be about 4 additional sites opening up, and as those open, they'll be listed as well. Next slide, please. And just to say a little bit about the study itself. As I mentioned, you'll be on the first double-blind placebo-controlled portion. Some participants will be getting ASP0367. Others will be on this sugar pill placebo. 2/3 of the subjects will be on the active drug at the top. About 1/3 of the subjects will initially be on this 12-week period, first on a low-dose period for 2 weeks and then a high-dose period for 10 weeks. And so there'll be this 2-to-1 randomization, essentially. And then after this portion, all participants will have an ability to choose whether they want to receive ASP0367 for another 3 months. So you'll have an opportunity to receive the drug for 6 months for some people, and for some people for 3 months. There'll be a 4-week follow-up as well. And I also want to mention about half of these visits have been converted into visits from home, just to make things a little bit easier during the COVID situation. The entire study takes about 8 months. If you choose to only participate in this double-blind, placebo-controlled portion at the beginning, it would take roughly about 5.5 months to participate. Next slide, please. So who can participate in this trial? It involves boys that are ages 8 through 16 that have a diagnosis of Duchenne muscular dystrophy, that are taking a stable dose of corticosteroids for at least 6 months, have at least some trouble walking as assessed by a 10-meter walk test in the first screening visit, and can also move their arms at least up to the shoulder height as well. And exclusion criteria include having an acute illness recently, or some serious infection, or mental health condition or behavioral problem, or is not healthy enough to be in the study at the judgment of the site principal investigator. Also, if you have certain heart problems, an ejection fraction less than 55% means that your heart has some trouble pumping blood throughout your body. A long QT interval means that there are some electrical conduction problems that your heart has. Or high levels of cardiac troponin, TNL, TN1 on a lab test. That's sort of indicative of potential muscle injury for your heart. So these would be exclusionary criteria. Also, having used certain drugs within 4 weeks of the study, there are some potential drug-drug interactions that we haven't fully tested, so we're being on the cautious side and excluding those for now. And also, if you have certain kidney or liver problems, or if you're currently receiving an investigational drug. And you can refer to clinicaltrials.gov with the ASP0367 that you can punch in to get more information about this as well. Next slide, please. In terms of study updates, there'll be a few that are changing. Initially, when we started this study, boys on commercial exon skipping drugs were not allowed, but we are actually adding that in amendments. Some have been processed by the IRBs at the sites at this time. There are few that are in the process of finishing that. But also, if your family has previously had COVID and now healthy, or the caregivers have been vaccinated when the caregivers are at high risk for COVID, this is another amendment that is being processed right now. So the site teams are working to implement the protocol amendment very soon. Some already have. And if these changes will apply to you, you can ask your site when the amendment will be ready for you to enroll. Next slide. So how are we mitigating coronavirus SARS-CoV-2 risk? So fortunately, as you can see in the news every day, the COVID-19 epidemic is certainly getting better here in the United States. There's always the danger of problems arising just given with the nature of the pandemic elsewhere in the world. We have taken a number of safety measures, however, to minimize the risk of infection with coronavirus. That includes, as I described earlier, home health care visits. We've changed about half of the visits to home health care visits, where nurses are specially trained into things that they can do to make sure they're not increasing the risk of you or your family members. They come to the home to do certain of the tests. Your study doctor will ask about your family's work and travel history to assess your recent or potential ongoing heightened risk of exposure to coronavirus. And also in terms of eligibility for the study, we've added some eligibility criteria to exclude people who have signs and symptoms of coronavirus or have unvaccinated parents or caregivers who are at higher risk of exposure due to recent travel, certain types of work, or having been near people infected with the coronavirus. Next slide, please. And so what happens at the study visits? There are a number of tests that are done, such as just looking at your general health. The study doctor will be examining you. There'll be blood urine tests as well. There'll be certain heart tests, such as the ECG, and we'll be looking at certain things coming from your heart, enzymes in your blood as well. And if there are any issues with those, then you'll need to get an echocardiogram as well. Echocardiogram is, as you're aware of, is just simply basically using sound waves to assess how the heart is functioning. Also, there are breathing tests. Pulmonary function tests are performed. If during the COVID issue, that we give that some discretion to the sites in terms of if they don't feel like it's needed to be performed in your particular case. We're also looking at how you move your body. There's a test in the clinic, a cycling test that I'll describe in a little bit. Also, we use the performance of the upper limb, 2-minute walk test, and some tests at home, such as video assessments and an activity monitor. We'll also be looking at how your body uses the drug, looking at drug levels in the blood and urine, and how you feel and think. There'll be some quality of data surveys, some diaries, some patient and parent interviews, a digit span memory test, and others. And we'll also be looking at muscle structure. About half the sites that will be involved in the study will be using a magnetic resonance spectroscopy. That's not up yet, but I'll describe that in a little bit as well. But first, the assisted 6-minute cycling test. In the next slide, this assisted 6-minute cycling test is basically analogous to the 6-minute walk test. It's measuring a person's endurance for the upper limbs, whereas the 6-minute walk test measures your endurance for your lower limbs. This outcome measure is used for people with DMD who are likely to stop walking soon or already require a wheelchair or power chair. To do the test, the person cranks the handles of the tabletop mobile trainer for 6 minutes, and the mobile trainer gives some assistance so that the handles are easier to turn. And the endpoint for this study is that we're actually counting the number of times that you're able to crank the handles with your arms over a 6-minute period. Next slide, please. And I also mentioned that at half the sites will be looking at muscle magnetic spectroscopy, or MRS as it's also known. So MRS is very much like MRIs. Both of them use sort of magnetic resonance imaging, and they're using magnets to take pictures inside the body. So it's the non-invasive imaging technology that's been used to study different organs such as the muscle or the brain. We're not using any radioactive compounds that you get when you're using like a PET scan or a positron emission tomography. So it's a non-invasive imaging technology. The person lies down on a rolling table, goes into the machine, they lie still, and then the machine uses magnets then to create the images of your body. In this study, the MRS will be used to create images of the muscles in your legs. And the amount of fat versus healthy muscle will be measured, which is called the fat fraction. This will allow us to assess how the disease progresses without doing any surgery or biopsy, really a biopsy. It makes it sort of a non-invasive way of assessing your muscles. And we'll be using this MRS to see if the ASP0367 might slow down how quickly fat replaces muscle because of Duchenne muscular dystrophy. And the next slide, slide 17, in terms of how can you learn more, as I mentioned before, you can visit www.clinicaltrials.gov, and you can search for NCT 04184882 for more details about this study and see a list of locations that are recruiting participants. And sites will be added once they're ready to accept new participants. And you can also email Astellas Pharma Global Development using astellas.registration@astellas.com, or you can call 800-888-7704. And also when you go to www.clinicaltrials.gov, you can type in ASP0367 when it asks for associated drug, and then you can find this ongoing clinical trial that way as well. So a couple different ways to reach that information. So thank you very much for listening to this presentation. I'm more than happy to answer any questions that you have that I didn't answer to your satisfaction.
Yeah, thank you, Dr. Merrick. This was terrific, and I'm going to take you back through the presentation and ask you a couple questions. I also want to say before we start the questions and sort of going back through, that if you're in the registry, the Duchenne registry, that's a very good place to learn about trials, because we do recruit through trials for sponsors who are interested. So you would, if you are eligible, you may get a notice from the Duchenne registry. In addition, on the PPMD website, there's a trial finder that's quite specific and we find clinicaltrials.gov can be a little confusing for many and hard to sort through. So those are two other options to find information about the study. So let's go back and start again, and we'll just go through some questions. On slide 3, if you can go back there, Kaylin. So this is slide 3. I don't know if you have this up, and I know your slide deck was freezing a bit, Dr. Merrick, but you talked about, first of all, combination therapies, and I just wanted to make a comment about that. I think this community is well aware that combination therapies are going to be needed in order to stop or dramatically slow disease progression, so we're really thrilled about that. And you mentioned that you've listed the sites in Sacramento and Kansas City and Massachusetts and Virginia, both sites, and you said 4 more sites are coming, just to clarify. And when you said coming, time is a commodity here in the world of Duchenne. So can you give us a sense of what time we're talking about? Are we talking a few weeks? They should be up and running by late June, or are we talking about a longer period of time?
We have a couple sites. Minnesota has opened up, so we have 6 that are open right now. We have another couple sites that should be opening within roughly a month, and then another site should be opening soon after that, and then a final site in Florida will probably be opening hopefully a few weeks after that.
Great. So
by
we hope
summer,
to have
all of them open up in a couple. Yeah.
Okay, that's really wonderful. And then go to slide 4. Thanks, Kaylin. This is when you just gave us the overview of Astellas, and I just have sort of a out of the blue, just because we are familiar with the Japanese companies as they both, as they were consolidated into Astellas. I wondered with this compound, were there any safety studies done on this compound in Japan?
There haven't been any
prior to starting a trial.
Yeah, there haven't been any clinical trials done in Japan yet, but that obviously will be something that we'll be looking to do once they'll probably be dependent in part on the results from this study, yes.
Okay. Thank you. Sometimes we see companies that start on safety in their in the company of origin and then come to the U.S. So it looks like this is going to be the reverse, U.S. first and then back into Japan. Slide 5, please.
Right.
This is where you talked about the biology of the mitochondria, and I just wanted to reinforce, this is, when I was long ago in school, we talked about the mitochondria being the furnace of the cell and providing all the energy for the cell. Just to clarify, in Duchenne muscular dystrophy, the mitochondria are fewer in number and damaged. Is that both of those things are true? Is that correct?
Yes, it is, yes. And
right, so the mitochondria...
Yeah, go ahead.
Sorry, go on. No, go on.
So
just that they are damaged. Yeah.
Yeah, and our hope is that this, by actually acting to make the energy production by the mitochondria more efficient, this will help actually sort of the cells will be a little bit more resistant to that damage of calcium entering, and then it will also increase the number of mitochondria as well, because that's an effect that we see with this drug in animals and human cells as well, yes.
Yeah, thank you. That's what I wanted to get at. Not only is it going to hopefully repair the mitochondria that exist that are damaged, but hopefully increase the number of mitochondria within the cell to help with the energy of the cell. So that's good. All right, slide 6. This is a PPAR delta. This is where your drug interacts, and you talked about other drugs that really have an impact earlier than this. Can you just describe what drugs those are that have an impact earlier, and is PPAR delta used alongside of those drugs? Can you just sort of enlighten us a little bit there?
The ones that I was saying that also act on PPAR, there are a number of drugs for diabetes, so obviously it's working on energy production there. Those drugs are acting on a little bit different PPAR site, though. And this is actually a delta pathway that's actually unique. So that's why the critical thing is that we're having to look at this safety for our compound, because just confirm that There are some issues with other PPAR drugs. In animals, we don't see those same kinds of issues for the PPAR delta pathway, however. And so, the important point is that this PPAR delta pathway is part of what's actually activated on the mitochondria and turns on the genes within and regulates the mitochondria in that manner.
Yeah, so in Duchenne, we often hear from some of the young men and even some of the carrier moms, which is a different topic, obviously, and not included in this particular study, that fatigue is really significant in them, that they are really get worn out quite quickly. I know some of the little boys have complained to their parents about my legs feel tired. So you think this has to do with the energetics of the cell, and this is why the approach with the PPAR delta.
No, exactly, and that's one of the things that we're certainly interested in testing. Not only that you can do more in terms of muscle performance, in terms of time tests, for example, but also that subjectively that we're able to alleviate some of those feelings of fatigue or soreness in the muscles, yes.
Good, and then and I'll just put a plug in for carriers, and I know that's not your focus, but should you see success here, I think some of the female carriers certainly experience a great deal of exhaustion and fatigue in muscles. So that might be an idea later on down the road as you see your success.
Yes.
All right. Let's go to slide 7. So I think it's terrific that you're testing a combination therapy. So you're testing the antisense oligonucleotides that are currently on the market, which is skipping 51, 53, and 45. So that's very interesting and exciting. And then people who are on a steady dose of steroids or steady state steroids. Can you talk a little bit about steady? I know steady state means anything, and heaven knows there's a lot of different regimens that are being utilized in Duchenne. But is there a preference in terms of the study that you would think that the drug might interact better with daily steroids or intermittent steroids, or have you thought that through, or not?
Our plans are to actually allow both like daily taking of steroids, like probably most boys do, but for those that, for whatever reason, working with their doctor, they've decided to do like a regular weekend treatment with the steroids. We're going to accept both, and so that will provide us with an opportunity to, at least it'll be small numbers, but to at least understand safety and tolerability. Certainly, yes. So we'll be looking at both, allowing to look at both of those. Yes.
Good. And then in this particular slide, you said some approved DMD drugs act upstream of the ASP0367. You're talking about the antisense drugs, right? That they act upstream,
well, yeah.
And then you're hoping this is complementary or synergistic with those drugs.
Yeah, essentially ASP0367 would be acting downstream of dystrophin, so those drugs would be acting upstream of where ASP0367 acts in the mitochondria.
Exactly. All right. Good. Thank you. And then let's go to slide 9 and talk about the trial. So it's a phase 1B randomized. Sorry?
Go ahead, yeah. Go ahead. Sorry.
Okay. Phase 1B double-blind placebo-controlled trial. How many boys are you recruiting to this study?
We're recruiting 18 boys to the study, so 12 will be on ASP0367 initially during this double-blind portion, and 6 will be on placebo during this double-blind portion. And after we finish that, there'll be an open-label period for 3 months duration, in which everyone would be on ASP0367.
Good. Go to the next slide, Kaylin. Yeah. Okay. So that's terrific. So it's an 8-month study, and I think the next question that every parent will ask you is, at the end of 8 months, what happens next? Are you planning, if you see safety and you see something there, will you plan an extension study, or when will you make the decision about whether to extend it in these 18 young men who participated in the study? I think it really is, if you're taking a drug that is safe and you feel that there's benefit, and even if you have a trend toward benefit, to discontinue the drug is really hard and very difficult to accept.
Right, yeah.
So are you thinking of an extension phase?
For this study, we actually won't have an extension phase. We could rethink that if we see very, very clear, a very clear effect in this study. I mean, it would sort of need to be a very, very robust effect, and then we would change our minds. If we were convinced that there's clear efficacy, then we would be in a position to offer that. Since we haven't really clearly demonstrated efficacy yet, we don't feel that we can offer that, and would want to make sure that boys would be able to participate in some additional trial where they would have hope for seeing effects with another agent. But I understand the issues, and when we do our next study, we will plan on a longer open label period so that we have an opportunity to really address and see what ASP0367 is doing. And then if we have clear signs of efficacy, we can continue treatment on for everyone, yes.
Yeah, I just think for these 18 young men in the study, right, there should be a forward-looking plan that says we're going to do a follow-on study when we see this. Just because I think making a decision to join a trial, it's really important that you're fully informed about what the trial is and what your expectations should be. You're going from low dose to high dose, and obviously doing some outcome measures that we'll talk about in a minute that are new for our community. So I think making sure you inform these parents and these young men about your plans to do another study should you see benefit, or that you're going to look at a certain point, and if you see robust benefit, that we won't leave these people out of the next study or just say thank you for being in the study and then with no plans to go forward. As you know, we all need combination therapies, and so I want you to keep that in mind.
Right, yes.
All right. Okay, so if we go to slide 11. So just a little clarification of this. When in the inclusion criteria, 8 to 16 is fine, stable dose of steroids, and obviously you'll add to that it's probably a stable dose of antisense oligonucleotides, so they can't just be starting the day of that. So,
right, yeah.
How long will they have to be on the antisense drugs? Have
we
you thought about that?
We would be looking at a period of 6 months there as well.
Okay, for both. Okay. And then has at least some trouble walking in the 10-meter walk test. Can you maybe enlarge on that a little bit? So you have to be able to walk 10 meters consecutively?
If you can't walk if you can't walk 10 meters and are not currently non-ambulatory, you're able to come into the study as well. So it's what we're really sort of looking at is if you're sort of on a somewhat later ambulatory or non-ambulatory, unable to walk that distance, and so there has to be sort of a clear, measurable effect, though, to be able to enter the study.
In ambulation.
Yes. Yeah.
Oh, okay. So you have to be ambulatory and able to at least move from room to room.
No, you don't have to be. I'm sorry, you don't have to be ambulatory, but you need to have some difficulty with ambulation. Yes. Okay.
So the minimum is difficulty with ambulation. So if you're a fast walker and can do just fine, that's not part of the inclusion criteria. So you have to be at the point of losing ambulation. Yes. Okay. Yeah. So I just wanted to clarify that because I found it a little bit confusing. So I'm glad you did. So this study is for a person who's just about to lose ambulation, can maybe move a little bit on his own, but certainly on the brink of stopping to walk, right?
Right. The inclusion criteria were largely sort of written to try to avoid being, we're trying to recruit the same subjects, same participants that many of the genetic therapies are currently trying to do. Although they're starting to look at later stage in some non-ambulatory patients now. But it was sort of meant to sort of avoid the issues of having a difficult time recruiting because so many people are and families are interested in those genetic therapies right now.
Sure. So this is really looking at transition.
Yeah.
And here's another question, and I'm sure the answer is no, but I'm going to ask it anyway. If you have participated in a gene therapy and you're 1 year out, 2 years out, is it possible to be considered here, or is the exclusion criteria excluding people who've been in an AAV-based therapy?
Yeah, the exclusion criteria that you're currently, you're receiving an investigator drug would sort of apply that you've had an investigational drug, and that's something that hasn't been improved. So for this study, we would be excluding such subjects. Down the road, we're going to obviously have to address that issue, though. So because I think it's probably not reasonable to continue to exclude those subjects permanently, because obviously we sort of view it as a compound like this would be ultimately, we would envision it to be something that could be used even on top of people who had gene therapy, because the expectation is that it's going to be a number of different types of treatments that are going to be needed for every boy to sort of maximize their function properly.
Right, and I think just thinking through the gene therapy, while it is an investigational drug, it's not something you can withdraw from, right? So
right,
we have
exactly,
to think
yeah.
very clearly about this population of boys at this moment who've had a gene therapy, because once they're a year or two out, I mean, they're no longer in the, they're being followed, but they're no longer per se in the study, depending on how long the follow-on is going to be. And so I think if we're
right.
thinking complementary therapies, we have to think through including these individuals at some point in time. And as you know, time is the commodity here. So I think that should be on everyone's radar as they develop drugs, because yes, investigational, and if it, we're all hoping gene therapy is approved, but if it's not approved, then that would eliminate this person because they're still on an investigational drug for the rest of their life, which is certainly not fair. So
right.
As I say, we're all hoping for approvals, but we have to think through how can we include them. So you also talked about certain other type of drugs within 4 weeks. Can you tell us what those other certain type of drugs are? Are we talking about coenzyme Q? Are we talking about idebenone? What are we talking about in these certain drugs?
We're largely talking about drugs that act on sort of certain liver enzymes that sort of are involved in metabolizing these drugs. So drugs are a foreign object coming into the body, and there are enzymes in the liver that have been developed over time to sort of to metabolize drugs and to break them down into smaller pieces. And so some of these drugs that are acting on a certain enzyme called CYP3A are ones that we actually have to minimize. And so there certainly will be some drugs that this will be relevant for, but there will be many that it won't, that are not there. I don't think we're going to be losing a lot of boys, participants, for this reason. But it is one of the exclusion criteria that we have at this time. Down the road, we'll be doing formal studies looking at drug-drug interactions, and then we'll be able to open it up a little wider. But we haven't had a chance to do those type of studies yet at this stage of development.
Yeah. Can you give us an example of one of those kind of drugs, just so we have a hint, so a parent doesn't decide to screen to find that they're on a drug that has to be discontinued?
Well, like some of the worst there are certain antifungal drugs, for example, that this is true of. Okay. There are a range of drugs that are used for the treatment of HIV, for example, that this is true of. So, those
probably
are
not
some of the range that would not be involved in the population here. Yes.
Okay, great. All right, let's go on to slide number 12. So first of all, this is terrific, and how to enroll. And we're all very acutely aware of the pandemic and what seems like the world that's closed down for more than a year. So when you talked about the fact that people and their caregivers should be vaccinated, I just have a question to you about these young men that are now able, especially in your age range, to get the vaccination. So is there a window between receiving that vaccination and screening for this study? I know in other studies there's a 30-day window, so you need 30 days before you screen for the study. And I don't want people to not get the vaccine because they're trying to screen for the study. So can you give us a little bit of color on getting the COVID vaccine and still eligible for participation, but you need a window or not?
Yeah, we actually don't have a window built in because we're for just given the nature of the COVID crisis, we're certainly not wanting to discourage anybody from getting vaccinated at all. We would prefer that there be a pause of something like about a month. That would be ideal, but we're not formally requiring that. Yeah.
Okay. And then in the next slide please, 13. You mentioned the unvaccinated caregiver. That's a pretty sensitive subject. So how will families know, or how will you know that they might have a vaccinated caregiver, an unvaccinated caregiver for the exclusion criteria?
That would really be coming from the discussion either in screening before you, and just actually in the first screening visit with the at the site, and that's largely where that would come. Yeah.
Okay, great. Next slide, please. 15. All righty. So I think this device, the A6MCT, is probably new to most families. This is a arm test as described by Dr. Merrick, and this was really developed by, I believe, Imelda de Groot in the Netherlands. Is that right?
Yes, absolutely.
So Imelda is a physician, physiatrist, rehab doc in the Netherlands who worked on this and really compared it to the 6-minute walk test. I think that this is really done well in Europe, and I think it should provide some very useful data. So each family would have one of these devices at their home. Is that right?
Well, they'll have one of these at their site. So we're not testing it every week. So we're actually only testing the performance of this actually at the clinical site. And so the physical therapist will be with them as well, just to try to standardize this a little bit more. That was certainly one of the things that we thought about, we would have sort of loved to have done. But this is one of the nice added features of this study, though, is that people that would be involved in this study, they're also helping us provide additional data for the 6-minute cycling test, because there aren't as many upper limb function tests as we have for lower limb function tests. So it would be great to further validate and understand have a greater number of tests to choose from for assessing upper limb function as opposed to the range of ones that we have for lower limb function now.
Right. So thank you for that explanation. So for the group on the phone, the Brooks score is the first upper limb function that was developed, and basically it is scored on how well you can get your arms above if you can hold both arms above your head, that's a perfect score, and then it's incremental in other measures. And to find a more sensitive test, a group of people, patients, families, advocates, physical therapists, etc., developed the pool or the performance of the upper limb. The pool divides the arm into 3 different domains: the shoulder domain, the elbow domain, and the wrist domain, and tests there, and that is also scored. And then Imelda developed this that she believes is even more sensitive and more comparable to the 6-minute walk test. So this is one of your exploratory measures that you'll be seeing sooner and probably more frequently, as we really want to involve boys who are in transition or non-ambulatory, and certainly adults who are non-ambulatory, in studies where there is the hopeful opportunity to participate and to see the development of a drug that can improve or preserve their upper limb function. So thank you for that. I think that's really terrific, and I appreciate that you've decreased the burden on this. I think that's the next slide, 16 or 7. Yeah, 16. And this is MRS. So a little bit, Dr. Merrick, I think many on the phone are probably very well aware of imaging DMD, where they are doing an MRI of the lower leg or the whole muscles. And the MRS is a little bit different, and you're talking about certain sites will have the MRS. Can you add a little color of what the MRS, it's less burdensome in terms of the way it works than an MRI, perhaps. But, as we see these boys that are in transition, they may have contractures in the hips. They may have contractures behind the knee of that hamstring. So are you still able to do an MRS on those kinds of, on people with those kinds of issues?
I can't speak to all of them in particular, but we're going to be using largely sites that have been really working with involved in working with imaging DMD for a while. So my understanding is that the technicians working at these sites are really used to working with this population quite a bit, and so I think that they're going to be in a position to deal with many of these cases. Certainly, yes.
Good. So we are all familiar with imaging DMD, and certainly for the group on the phone, they are working to, the fast action is validated, and they're looking forward to it as a biomarker in Duchenne and working with the FDA to qualify that as a biomarker. So I think this looks like a very good study. Last slide, please, 17. Dr. Merrick, I think this is a very interesting study. We're very excited about improving the energetics of the cell, which hopefully will improve the energy of the individual. We're really thankful to Astellas for taking this forward. We knew the company MITOBridge long ago, and we certainly are very anxious to see this come into the clinic and clinical trial, and we're very happy and willing to see this go forward. So we really appreciate your time today and your explanation. And for those of you on the phone, please seek out the Duchenne Registry, the Clinical Trial Finder, clinicaltrials.gov, or contact Astellas as outlined here in the 800 number. This is a very interesting trial, and I think really thinking through combination therapies, how can we improve the energetics and build upon those antisense oligonucleotides or the steroid use, or someday soon gene therapy. Thank you all for listening, and thank you, Dr. Merrick and Astellas, for taking time to explain your progress in Duchenne muscular dystrophy. We'll see you all again soon. Have a really good day.
Okay, thank you very much.
You're very welcome. Thanks for joining us.